wonderful tour of the different areas of the lab. Now let us see what are the different instruments and equipments we have in our laboratory. So the first instrument what you can see is the cyclomixer. Now this cyclomixer is used to mix different reagents, maybe solutions, maybe bacterial suspensions, so as to create a even solution. The second instrument, what you can see, which is a huge and a large instrument, it is a cooling centrifuge. Now, there are some experiments like RNA extraction, DNA extraction, where the temperature needed is not room temperature, but maybe 4 degrees or 5 degrees. So as to achieve centrifugation at such temperatures, we use this cooling centrifuge. Then the third instrument, what you can see over here, it is the incubator. Now on the incubator, you can also see that the standard operating protocol, also called as SOP, is being also mentioned. Okay, so let us see what is inside. Okay, so over here, this is the incubator where the teachers or maybe students keep their plates, flasks which are with the cultures for incubation, so that the bacteria which is present, these will grow and multiply. Then the next instrument what you can see over here is called as a rotatory shaker. Now over here, this rotatory shaker is used to incubate, or it is used to grow the cultures which require aeration, that is oxygen, as well as room temperatures to grow. Then the next instrument what we have is one of the unique instrument. It is UV visible spectrophotometer. Now this UV visible spectrophotometer is very essential to use for maybe quantification of DNA or RNA sample, protein sample, or to determine maybe the nanoparticle intensity and different other solutions. So let us see. So I am just opening the case. It is very well protected. So this is the UV visible spectrophotometer. And over here, you can see the instruction sheet for the module of the UV visible spectrophotometer. Then how to do the carry out the calibration curve of spectrophotometer is also given here, as well as the standard operating protocol of UV visible spectrophotometer. So these are the instruments which are being present in the instrumentation area, which I had already showed you in the last video. Now the rest of the different equipments and instruments what we have. So over here, the one of the instrument which you can see, it is called as a horizontal gel electrophoresis. Now this horizontal gel electrophoresis is being used to carry out agarose gel electrophoresis, that is to separate the DNA. So this is how the instrumentation looks like, and this is the power pack. Now as we move ahead, we go over here and we see a second instrument or equipment which is present. This is called as the vertical gel electrophoresis apparatus. Now vertical gel electrophoresis apparatus, this is a larger version. So this instrument is used to separate out the proteins. Then a smaller version of the vertical electrophoresis is being displayed over here. So over here, this is used for a small sample size, while the previous one was used for a large sample size. The next instrument what you can see, it is the water bath. Now water bath over here is required for some experiments where heating is required or maybe digestion of samples is required. So over here one can have a temperature range of as low as maybe 20 degrees Celsius to as high as 110 degree Celsius. Moving on ahead, we move to next particular instrument, which is the colorimeter. Now the colorimeter over here is used to measure the absorbance or the optical density of a sample or of a bacterial culture, which is provided with different filters, as you can see over here. The next instrument, it is called as a microfuge. Now you have heard of centrifuge, but yes, we do have a microfuge where we want to centrifuge the samples which are very low quantities, like maybe less than 1 ml for analysis. Then we have heating mantle. Now this heating mantle is used for some you can say experiments for which heating has to be carried out in maybe for research purposes or for any you can say extraction purposes of a metabolite from a plant sample or maybe from any you can say solvent. Then for that heating is required of very high temperatures. So this particular heating mantle is used for the similar purpose. Then we have UV cabinet. Now this UV cabinet is used to observe those samples which fluoresce under UV light. Okay, for example, for thin layer chromatography analysis. So over here you can see this is a chamber wherein you can put a particular samples, and from here one can observe. Now over here one can see there are 3 knobs, that is short, ultraviolet, long ultraviolet, as well as visible light. What you can see, it is the magnetic stirrer with a heater. Now there are some reagents or there are some chemicals which do not dissolve easily in liquids or maybe in some solvents, or they take very long amount of time. So manual particular stirring or maybe manual mixing will take a lot amount of time. So to reduce the time, this magnetic stirrer is used. This is UV visible ultraviolet transilluminator. Now this ultraviolet transilluminator is used to observe the DNA samples which have already been separated by the horizontal gel electrophoresis. And this is the microscope, which is most commonly used instrument for observing bacteria, maybe some tissues on a glass slide. And this is the weighing balance for maybe measuring some chemicals, some maybe components of media, or maybe measurement of some samples. For this, weighing balance is used. In the discarding area, where we see the next instrument, which is the autoclave. Now this autoclave, what you can see, it is a large autoclave or a big autoclave. Okay, this is a medium size autoclave. Yes, we also have a small version of the autoclave, which is this called as a small autoclave. Now all the 3 autoclaves which are being present are used for sterilization purposes, but specifically this small version as well as this medium version is used for sterilizing or for preparing for sterilizing the media, while the 2 which you can see in the next room is used for discarding purposes. You can see it is the steam distillation unit or distillation water unit. Now this steam distillation unit or distillation water unit is used to distill the water so that distilled water can be made. Now this distilled water is used for many purposes, like preparation of media, or as a you can say diluent, or maybe preparation of saline, or maybe for other purposes. What you can see, it is the hot air oven. Now the hot air oven which is present is used to dry off or to clean dry off the materials or glasswares which are present. So as we go inside. So we see there are different glasswares which are being present, which have been kept for drying. Okay, and over here you can see there are pipettes also which have been kept for drying. Culture lab, we see that again here it is the shaker incubator, but this shaker incubator is different from the one which you've already seen earlier. Here the shaker incubator is used for those bacterial cultures which require 37 degrees Celsius to grow, but at the same time they require aeration. So let us see what is present inside. So I'll open the second door, and over here you can see that it is the same general construction as we seen of the rotatory shaker. Is the laminar airflow, which is the horizontal laminar airflow. So this laminar airflow over here is used to conduct those experiments which require strict aseptic conditions. Okay, as for other experiments like to study the effect of UV light on maybe cells, maybe bacterial cells. So for such experiments, this laminar airflow is being used. Commonly used particular instrument is this centrifuge. So centrifuge is used to separate those components from maybe a mixture of you can say the solid component from the liquid media, or maybe precipitation purposes, or for other you can say purification of some compounds. For those purposes, this laboratory centrifuge is used.